So now that you have an understanding of how each individual muscle cell shortens, it's time to move up a level and look at how this relates to the action of the whole muscle. We'll begin by looking at the length and tension relationship. The amount of tension that's generated by a muscle, and therefore the force of its contraction, depends on how stretched or contracted it was before it was stimulated. If it's overly contracted, we have a weak contraction. If it's too stretched, we also have a weak contraction. So there's an optimum resting length for the muscle in order to produce the greatest force. If we look at the diagram over here on the right, we can see how this tension-length relationship works. When the muscle is overly contracted, then we see that there's no space for the myosin to contract the sarcomere any further. When the sarcomere is overstretched, then very few myosin heads are able to interact with the actin filaments, and thus there's not that much power in the contraction. However, at an optimum resting length, we have both space for the myosin to walk along the actin filaments, as well as enough myosin head interaction to generate a strong contraction. So when you think about this, working out in the gym perhaps, you see people often working just in their optimal resting length zone. They'll use heavier weights and not go to the full extension of the joint or the full contraction of the joint. They'll gain much more strength benefits if you go through the full range of motion with a lighter weight, because we're able to get a deeper contraction here, and we're able to train the muscle while the myosin heads have very few interactions, which causes them to develop more interactions. So much better to work with a lower weight and the full range of motion rather than a higher weight that can't be maintained using a full range of motion. So in many labs, we study muscle contraction using the gastrocnemius muscle of a frog, which can be easily isolated from the frog along with its sciatic nerve. This preparation can be attached to a stimulating electrode and a recording device that produces a myogram, a trace on a chart, giving the timing and strength of muscle contraction. A sufficiently weak subthreshold stimulus will fail to cause any sort of contraction. It takes a threshold or minimum voltage to actually generate an action potential in the muscle fiber to produce a contraction. The action potential naturally triggers the release of calcium into the cytosol, which activates the sliding filament mechanism. At threshold or higher, this causes a quick cycle of contraction or a twitch. This figure on the right illustrates this. The time of stimulus and then a contraction. If we reach this threshold level of stimulus, then the contraction can happen. There's a latent period or a delay between the onset of the stimulus and the onset of the twitch response. This is the time that's required for excitation, contraction, coupling, and tensing of the elastic components of the muscle. The force generated during this time is called the internal tension. It's not visible on the myogram because it doesn't cause any shortening of the muscle. Once everything's become taut, the muscle begins to produce some external tension. This is when we're in the contraction phase. And the actin and myosin filaments slide across each other and the muscle shortens. During relaxation, the sarcoplasmic reticulum quickly reabsorbs the calcium, the myosin releases the thin filaments, and the tension declines. The muscle is going to return to its resting length. The entire twitch lasts from only 7 to 100 milliseconds. It's very fast. Here we can review again each of the phases. Time of stimulus, a short latent period, then we have the contraction, maximal contraction, and relaxation. Very, very short period here on the time. A subthreshold stimulus won't elicit any contraction at all, and anything above threshold is not going to elicit any greater contraction. It's an all or none phenomenon. where contraction is at its maximum or it's not there at all. When we actually look at a whole muscle, this all or none phenomenon is not necessarily true. The twitches vary in strength depending on a few different factors. First of all, the stimulus frequency. If we have many stimuli arriving close together, it produces stronger twitches because there's simply not enough time for calcium to be returned to the sarcoplasmic reticulum. So the concentration of calcium in the sarcoplasm remains fairly high. And when there's more calcium in the sarcoplasm already, then the muscle already has developed some tension. A repeated stimulus will dump out more calcium and perhaps recruit more myofilaments. And we've investigated already how when a muscle is very stretched, it has less tension because there's less actin-myosin interaction. And when it's at optimum length, it will have a stronger contraction. Now, temperature definitely has a big influence. This is why we warm up before we exercise. A warmed up muscle contracts much more strongly because a warmed up muscle has all the ingredients there for contractions to happen. When the pH changes, like there's an accumulation of carbon dioxide and thus hydrogen ions, then we'll see that the contraction is weakened. This is a sign of fatigue. We've used up all the oxygen, we've produced a lot of carbon dioxide, and as you'll learn next semester, that ends up generating greater acidity in the sarcoplasm. Another huge factor is our state of hydration. If the muscles are not well hydrated, then it's going to affect the overlap of the thick or thin filaments. Imagine the size of a muscle cell when it's well hydrated versus the size of a muscle cell when it's not. This is one of the reasons that creatine phosphate actually works to develop more strength in a muscle contraction. Having additional creatine phosphate in the cells causes a cell to become overhydrated, which allows for generation of stronger contractions. So creatine does increase the strength of a contraction. However, this could be a bad thing, especially when it comes to the heart muscle. Now, it's not surprising that muscle twitches vary in strength. After all, an individual twitch is not strong enough to do any useful work. Muscles must be able to contract with variable strength for different tasks, such as lifting a glass of champagne compared with lifting a car. So let's look a little more closely at the contrasting effects of stimulus intensity versus stimulus frequency. Let's say we have one of those frog sciatic nerve preparations with the gastrocnemius muscle. If we give it a subthreshold stimulus, it produces no response. At threshold, we see a weak twitch. You can see that here at number 3. We get our first actual twitch. And if we continue to raise the voltage, we see stronger twitches. The reason for this is that the higher voltage recruits more and more motor units. As you can see here in this middle line, as the stimulus goes up, we get more and more motor units stimulated, and then we reach a maximum contraction strength. The process of bringing more and more motor units into play is called recruitment, or multiple motor unit summation. This is how the nervous system acts to naturally produce varying muscle contraction strengths. But even when the stimulus intensity remains constant, twitch strength can vary. And it varies with stimulus frequency, where high-frequency stimulation produces stronger twitches than low-frequency stimulation. We can see in this figure that when a muscle is stimulated at low frequency, it produces an identical twitch for each stimulus. However, when we raise it up to about 10 or 20 stimuli per second, we'll see that the muscle still recovers fully between twitches, but each twitch develops more tension in the muscle. This pattern of increasing tension with repetitive stimulation is called trep, or the staircase phenomenon. The muscle still fully recovers between twitches, but the stimuli arrive so rapidly that the sarcoplasmic reticulum doesn't have time between stimuli to completely reabsorb all of the calcium it released. Thus, the calcium concentration in the cytosol rises higher and higher with each stimulus, which causes the subsequent twitches to be much stronger. In addition, heat released by each twitch can cause the muscle enzymes such as the myosin ATPase to work more efficiently and thus produce stronger twitches as the muscle warms up. When we raise the stimulus to even higher frequencies, say 20 to 40 stimuli per second, each new stimulus arrives before the previous twitch is over, which causes a phenomenon called incomplete tetanus. Each new twitch rides piggyback on the previous one and thus generates higher tension. This phenomenon goes by two different names, either temporal summation or wave summation. Temporal summation because the stimuli are arriving closer together in time and are thus summed, or wave summation because one wave is added to the other wave. Each twitch reaches a higher and higher level of tension than the one before, and the muscle relaxes only partially between the stimuli. This produces a state of sustained fluttering called incomplete tetanus. So then why is it that our muscles aren't fluttering the whole time? The smoothness of our muscle contractions results because the motor units function asynchronously. That is, while one is relaxing, another is contracting, which makes it so that the muscle doesn't lose tension. Now let's say we were to increase the stimulus to 40 or 50 stimuli per second, which couldn't really ever happen in nature. We only get up to about 25 stimuli per second. It would form complete tetanus. This is not to be confused with the condition caused by the Clostridium tetani bacteria, but it does form this constant paralysis of the muscles because of continuous tension. In this situation of complete tetanus, we'll see that we have summation and incomplete tetanus until we reach a maximal level. The twitches then fuse to form a prolonged contraction, which we call a complete tetanus. A muscle in complete tetanus produces about 4 times the tension of a single twitch. Again, this would never happen naturally in our body. So this would be a great time to close your books, pull out a piece of paper, and see if you can distinguish between these different phenomena. Begin by describing a length-tension relationship. And then think about threshold, latent period, and twitch. What's the concept of recruitment? Differentiate between temporal summation, incomplete tetanus, and complete tetanus. And now we'll move on to look at isometric versus isotonic contractions. In an isometric contraction, we're having iso and metric, which means the same distance. So the muscle is producing internal tension, while an external resistance causes it to stay at the same length or even maybe become longer. This would be the example of trying to lift a car, virtually impossible for any normal human being. There's still tension developed in the muscle as we really try. However, the length of the muscle doesn't change because we're not actually doing any work and moving the object. Let's suppose you were to lift a heavy dumbbell. When you first contract the muscles of your arms, you can feel the tension building, but it's not enough to overwhelm the weight of the dumbbell. Your muscles are contracting at a cellular level, but the tension's being absorbed by all the elastic components as it's resisted by the weight of the load. Once we overcome the weight of the load, we move into an isotonic contraction, which is a contraction with a change in length, but no change in tension. This begins when the internal tension builds to a point that it overcomes the resistance. The muscle will shorten and move the load. Now, it maintains essentially the same tension, but as you know, there's a tension-length relationship, so it varies slightly. In order to have a true isotonic contraction, we would have to have a variable resistance. Isometric contraction is not merely a prelude to movement. It's certainly involved in our postural function, as the antagonistic muscles stabilize joints while we move the prime mover. When we look at isotonic contractions, we can have concentric contractions, which is where the muscle shortens and maintains tension, or eccentric contraction, while the muscle lengthens and maintains tension. Often in training, the eccentric contraction is the one that develops more strength. At the beginning of the contraction, we can see in this figure the isometric phase. We're developing tension in the muscle, however not moving anything yet. As we begin to overcome the resistance, we see movement, and so we move into the isotonic phase, where the muscle has the same tone, but is in a changing length. This has an inverse relationship to the muscle length. We can see the muscle length doesn't change in the initial isometric phase. However, when we move into the isotonic phase, we see that muscle tension remains the same, however, the length changes. So that brings us to the end of this section. Next, we'll move on to muscle metabolism.